Thanks for the introduction, and thanks for the invitation to be here. So today I'll talk a bit about some work that we've been doing over the last several years, looking at how individual differences in brain organization and connectivity are related to behavior. And a lot of this work, the overarching goal of a lot of this work is to try and develop what we're calling a stress test for the brain, and I'll get into what I mean by that. So before I dive in, I think there's two main reasons why we should care about individual differences. And maybe these are already obvious to some of you, but I think it's important to get on the same page. So the first major reason is that by studying individual differences, it can lead us toward a deeper understanding of cognition itself. So after almost 3 decades now of fMRI, we have these sort of canonical maps for different cognitive processes. So if we think about a working memory task or a language task or a social cognition task, we have sort of this constellation of areas that we all may think about. These particular maps come from thousands of subjects worth of data collected via neuroscience.org. And so this approach has taught us a lot about sort of general blueprint for human brain organization. But we also know that if we were to look at an individual subject or several individual subjects that go into these group average maps, we might see slightly different activation patterns for each of these 3 tasks or any other cognitive task that we could imagine giving people in the scanner. And so historically, it's been difficult to separate out just the noisiness of fMRI from something actually meaningful at the individual subject level. I think we're making really good progress in this area. But basically, the fact that we see these individual differences as people perform these cognitive tasks means that there's a degeneracy here. So in other words, there's multiple routes to sort of achieve similar or the same cognitive processes in different individuals. And this degeneracy could relate to a few different things. So it could relate to how people are doing on the task. And this might be related in sort of a linear, at least a monotonic fashion, such that more activity in an area makes people do better. There could also be nonlinear relationships here. But even if we hold performance constant across people, maybe people are using different cognitive strategies or styles to arrive at that same level of performance. And we also know that we can see some within subject changes, like with development, as we heard in several of the talks this morning, or even with learning on a shorter time scale. And so all of these differences across people could map on to the brain activity that we observe in lots of different ways. So there could be univariate or multivariate relationships here. There could be relationships with activity or connectivity or other features. But I think the main point here is that by trying to map directly from these individual patterns of brain activity to individual behaviors, this will get us a much more rich and nuanced understanding of cognition itself. The second major reason to care about individual differences is that it can give us insight into mental illness. So if we think about sort of a trait that exists on the spectrum in the healthy human population, for example, let's say we're talking about anxiety here. So we know that there's sort of this normative curve of anxiety where some people have less, some people have more, just on the trait level. The categorical view of mental illness is that we would choose some cutoff here, and we would say everyone that's down here on this end, I see my text is cut off, but that's meant to say patients. So let's say everyone in that red part of the curve gets a diagnosis. Everyone below that is called a control. And then typically when we do a brain imaging experiment, we would pre-stratify our participants into one of those groups. We would measure something about the brain, and maybe we see that there's sort of this mean difference here between the groups, but we also see that there's a lot of heterogeneity both within and across the groups. And so what I and many, many others are now moving towards is we're moving away from this categorical model toward a more dimensional model of these traits. So treating this trait as the spectrum that it truly is in a more continuous fashion. And so what this looks like when we do our brain imaging study is we would preserve that continuous variable, and we would try and relate something about the brain to these parametric levels of this trait. And so even in psychology studies where we may not have access to those stream values, if we can start to parameterize this part of the curve here, it may tell us more about what's going on at those extreme ends of the distribution. And the hope here is that by doing things in this way, we may be able to develop translational tools, so neuroimaging-based tools for real-world applications in the clinic, for example. So with those 2 major motivations in mind, I'll give you an outline of what I'll talk about today. So in the first part of the talk, ask and answer this question of, can we find individual differences in brain function that are robust and meaningful? Then we'll look a bit at how brain state impacts measurements of individual differences. Finally, I'll talk about some new and ongoing work looking at how we can manipulate brain state to draw out the individual variation that we most care about. Starting with this first section here, can we find individual differences in brain function that are robust and meaningful? And the primary approach that I'll be taking for the first two sections of this talk is functional connectivity. So I heard a lot about this morning, but just to review, and there's many different ways of doing connectivity analysis. The way that we typically do it is we take the brain and we carve it up into a set of nodes or regions. In this case, we're using 268 nodes across the whole cortex, subcortex, and cerebellum. And then for a given pair of these nodes, we can look at how strongly correlated their signal fluctuations are over time. In this case, we might observe that these two nodes tend to coactivate together. And if we do this for all possible pairs of nodes across the brain, we can get this symmetric correlation, or as we call it, connectivity matrix, telling us the pattern of how correlated activity in all possible pairs of nodes tend to be. And so we can calculate this functional connectivity matrix for a given individual doing a given task. And so the first thing that we wanted to know when we set out on this line of work a few years ago now is how stable is that functional connectivity matrix within an individual, maybe on different days or while the individual is doing different tasks. And so to answer this question, we turned to data from the Human Connectome Project. So back when this study began, we had access to 126 healthy adults worth of data from the Human Connectome Project. These were all pretty healthy young adults. Interestingly, there were 50 sets of twins in this data set. So 100 of the 126 people actually had a twin in this same data set, which served to make our problem even harder. And I'll get to what I mean by that. So the way that the data collection worked for this study is that people came in for 1 day of scanning. They did a resting state run, a working memory task, and a motor finger tapping task. And they came back on the second day, did a second resting run, a language task, and an emotional faces task. And what we did in this first study was we calculated those functional connectivity matrices that I showed you on the previous slide for every individual in every different task condition. And then we're asking the pretty simple question of, can we match up a connectivity matrix from one person acquired on 1 day to a connectivity matrix from the same person on the second day? And so we investigated all possible pairs of those scans. And to make a long story short, when we did that, we saw that accuracy was really, really high. So depending on the exact pair of scans we were using, we were getting between 54 and 94% accuracy with an average of 76%. And that was really striking to us because chance here is really low. So if we were just randomly guessing, we would expect to get this right 1 in 126 times. And instead we're seeing this really strong individual signal that seems to be persistent across these task states. And even when sort of the individual that you would expect to be most similar to you in the world, so if you have a twin, it might be that person, but we're still able to distinguish people based on these large-scale patterns of connectivity. And when we looked to see where in the brain the most identifying or discriminating power was coming from, we saw that it was these networks highlighted in purple and teal. So we're calling these sort of loosely the frontoparietal or medial frontal networks. These were the networks that had the most discriminating connections, and this makes sense given what we already know. These are the areas that are most recently evolved. These are the areas that we knew from previous studies show the most anatomical and functional differences across people. And so these were the regions that were really contributing the most to our identification results, which made some sense. Now this was interesting to us and exciting since it suggests that a lot of the variance in this functional connectivity is accounted for by who you are and not even necessarily what you're doing while being scanned. But the next thing we wanted to do was to see if these individual differences actually relate to some real-world meaningful output of the brain. So we wanted to see, in other words, if they related to a behavior measure that was required out of the scanner. And so everyone in this data set completed this test of fluid intelligence. So this is a progressive matrices task. Fluid intelligence measures meant to measure innate reasoning ability, sort of independent of learned knowledge. And so everyone, all 126 people in this data set did this task. And this is a task where performance does vary even among healthy adults. So you can see the histogram of scores in our particular sample here. So there's some variability. And we came up with a pretty simple sort of machine learning algorithm that we're calling connectome-based predictive modeling. And the idea of this is that we train a model based on n-1, so all but one subject's resting state functional connectivity data. And we find the connections that most relate to people's score on this test. And then we can test our model on that left out subject to see if we feed it in resting state data from a person that the model's never seen before, how well can we predict that person's fluid intelligence. And so when we do that, we can evaluate our model by correlating the scores that our model predicts versus the scores that we actually observe that people get. And so we see this nice correlation here suggesting that we can actually predict some, not all, but we can predict some of the variance in this sophisticated measure of cognition just based on patterns of brain connectivity while people are resting, doing nothing at all. And so this is a prediction here based on the whole brain connectivity pattern, but when we look to see which connections or which edges are contributing most to this prediction result, again, we see that it's our sort of frontoparietal temporal association regions that we previously saw were most discriminating of individuals. These are also the connections that best predict people's levels of fluid intelligence. And so since then, we've extended this approach to predict several different behaviors from functional connectivity data, much of it acquired at rest. So we've seen that this approach can predict, again, to some degree of accuracy, adults' ability to sustain their attention, as well as ADHD symptoms in children. We've seen that it can predict reading ability, as well as clinical symptoms of autism, even things like personality traits and creativity. So, I'll emphasize that these predictions, we have a ways to go before this would be ready to be rolled out in the real world, but we're seeing some encouraging evidence that there is some meaningful variability in just this resting state connectivity information. So to sum up the first section, can we find individual differences in brain function that are robust and meaningful? I'm going to say yes, so we can identify people based on their patterns of brain connectivity, and we can actually use these patterns to predict behavior. So at this point, you may be thinking, well, if we can get all this information from resting state data, so while people are lying in the scanner doing nothing in particular, does it really matter what we have them do while we scan them? And I'm going to say yes, it does matter, and I'll show you some evidence for that in the next section. So looking at how does brain state impact the measurements that we make. And to introduce this section, I'll walk you through a little bit of a thought experiment. So imagine that we have 3 individuals, harkening back to happier times, and these individuals are correlated with one another at some ground truth level of r equals 0.5. And in this case, that's actually just the correlation between the RGB values in each of these individuals, in each of these images, sorry. So that's the average level of correlation, but we can't measure that directly. We have to put people into the scanner and give them something to do, or not give them anything to do, as a way to measure the similarities and differences between them. So we could imagine a brain state or a cognitive task that makes these individuals look more or less similar to one another. And so if we think about a state or a task that makes people look more similar, we might end up sort of blurring out that individual signal. Here we're driving up the average similarity, but we're making it harder to actually recognize these individual differences. So maybe that's not what we want for studying individual differences. On the other hand, if we just kind of make people look less similar to one another in a completely unprincipled way, we can drive down that average similarity, but now we've actually destroyed a lot of the meaningful information about each person. So maybe what matters more is not so much the raw between-subject similarity, but rather this other axis here that I'm calling identifiability. And what I mean by that here is just how easy it is to look at one of these images and recognize who this person is. So we could think about a brain state that makes people look less similar to one another in a way that makes them more identifiable. So this is sort of this kind of caricature condition here. And then maybe somewhat paradoxically, at least at first, we could also think about a brain state that makes everyone look more similar overall, but does so in a way that preserves or even enhances the most important individual features. And so I just introduced this as sort of the general space that we're going to be looking at and trying to figure out where different brain states fall within this space. So to start with, we wanted to see if and how brain state affects between-subject similarity. So that's that horizontal axis from the previous slide. And so to do this, we turned back to HCP data, this time with an expanded set of 716 subjects. What you're looking at here along the x-axis are the 9 different functional runs. So these are 7 different cognitive tasks and 2 resting state runs. And the y-axis here is the distribution of all of the possible pairwise between subject correlations. So how similar are people's functional connectivity matrices calculated from data acquired in each of these 9 states. And these, what you can see here is some trends that make some sense. So for example, the brain state that made subjects look most similar to one another was this relational matching task. This was one of the more demanding cognitive tasks that the HCP ran. So that makes sense because if everyone is sort of seeing the same stuff and doing the same task, then that's probably going to make the connectivity look more similar. And then down here on the right side, associated with less between-subject similarity, we had the 2 resting state runs as well as the motor task, which was just a simple finger tapping task probably allowed for a lot of mind wandering. So again, the less constrained tasks are associated with less similarity, which makes some sense. But as I showed you previously in the sort of thought experiment, maybe what matters more for individual differences is not so much raw between subject similarity, but rather this identifiability metric. See that some of my text is flipped here, but that's okay. So basically, to get at this, we replicated the individual identifiability or the identification experiments. So this time with that expanded set of subjects. And what you can see here, so the way to read this matrix is that the 9 scan conditions are along the rows and columns here. And so each of these cells in this matrix is telling us how easy it is to match a matrix from one condition to the other. So for example, here we're matching the working memory task from day 1 to the resting state run from day 1. And what I'd like you to appreciate here is just that there's a lot of variability in this matrix. So these are all the exact same people that go into this analysis, but we're seeing a really wide range of values for how well we're able to identify people based on the exact pair scan that we use. And so specifically, if we look at our rest-to-rest matching, we see we're doing pretty well. So we're getting about 70% accuracy, but we're actually not doing as well as if we try and match people between 2 different cognitive tasks. So for example, if we match people between the gambling task and the relational matching task, even though these are 2 different tasks, we're actually doing better than at rest, which is in theory the same task both times. And so to relate this identifiability to the similarity values that I showed you on the previous slide, we correlated them here. So here what you're looking at on the x-axis is the mean between subject correlations. So that's just the mean of those box plots that I showed you on the previous slide. And we're correlating that against the mean identification rate. And so what that is just the average of each of these rows. So on average, how well does a brain state perform for identification when paired with all of the other possible conditions. And we're seeing a strong positive relationship here. And what that means is that brain states that make subjects look more similar to one another actually make better databases for identification. And that's a little bit counterintuitive maybe at the outset, because we might have thought that we want to sort of maximize between subject variability. But actually what we're seeing is that having some kind of task, so having some sort of constraints on the brain state that people are in, is actually getting us a better identification. And so the way that we're interpreting that is sort of having some kind of task on board to stabilize the functional architecture in some way that makes it easier to match across even 2 different tasks than it is to match across the resting states. And as a final analysis in this piece of work, again, maybe what we should be trying to maximize is not so much individual identifiability, but rather our ability to predict behavior. And actually, before I go on to those results, I'll just point out, so the 3 conditions that were associated with the highest between subject similarity and the highest in individual identifiability were these 3 tasks here, so the emotional, gambling, and relational matching tasks. And so we could think about these tasks as maybe falling somewhere in this part of our theoretical space. But again, maybe what we should be maximizing for is our ability to predict behavior. And so the last set of analyses in this part that I'll show you, we varied the input to our connectome-based predictive model to connectivity data from each of those 9 brain states. So those are along the rows here. And then we're trying to predict 3 different out-of-scanner behaviors. So the first one is that fluid intelligence measure that I presented before. We also tried a verbal intelligence measure and a working memory task, again, all 3 outside the scanner. Cells is the correlation between predicted and observed values. And I'll just say that actually all of these values are technically statistically significant, but you can see that the 2 resting state runs are actually doing the worst across the board for predicting behavior. And the tasks that are doing the best are the working memory and the language tasks. And so again, it seems that just having some kind of task on board, so even the motor task, for example, is giving us better predictions of fluid intelligence than just having people simply resting. And this could be due to any number of reasons. It could be due to data quality issues. It could be due to arousal. It could be due to a lot of different things. But I think the point here is that the tasks are actually giving us a lot more bang for our buck. Move on to the next section in the interest of time. But to sum this up, we're seeing that rest may actually not be the optimal state for studying individual differences. And it seems actually that the presence of a cognitive task can stabilize the functional architecture at an individual level in a way that allows us to better predict behavior. And so in the last few minutes, talk about some more recent work that we're doing to look at how we can actually manipulate brain state to draw out variation of interest. So given that we see these big state effects, can we actually tailor the brain state to the behavior that we care about to sort of elicit more signal to noise, so to speak, or better evoke the individual differences that we most care about? And so the trait that we decided to go after first was paranoia. And so paranoia is an interesting one because obviously in its extreme it's associated with schizophrenia and other psychotic illnesses. But we also know that paranoia varies even among otherwise healthier subclinical individuals in the general population. And the brain state that we created to try and study individual differences in paranoia was a narrative that was intended to be sort of ambiguous with respect to the level of suspicion that it aroused in different individuals. So the way the study worked was that people came in for 2 different visits. The first one was purely behavioral. They did some basic cognitive tasks as well as self-report scales, including our main measure of interest, trait paranoia. And then about a week later, they came back for the fMRI visit, where everyone heard an auditory recording of the exact same story. The idea was that individuals would interpret or react to this story differently depending on their baseline trait paranoia level. And unfortunately, my emojis aren't showing up here, but these are supposed to be 3 different emojis representing sort of different emotional reactions to this narrative. And so moving away a little bit from functional connectivity, the primary analysis approach that we'll take here is intersubject correlation. And so this is the advantage that we gain from having an actual task on board, and especially one that's time-locked across people. So now, instead of looking within brains, levels of functional connectivity between regions, we can actually look across brains and say, so given the same brain region, if everyone hears the same story and we see that activity in that region is correlated across 2 people, we can say that that region is somehow reliably involved in processing the stimulus across subjects. And in our case, we're going to take that a step further and look for regions that are responding more synchronously or more reliable in certain groups of subjects than others, and seeing if that varies at all with trait paranoia. And so before I show you the variation with trait paranoia, I'll just show you the intersubject correlation across the whole sample. So these are areas that show significant synchronous activity while people are listening to this story. So happily, some of our most synchronized areas were in primary auditory regions, again, because it's an auditory stimulus. But outside of this, we actually saw strong synchrony here in the temporoparietal junction, and if we do a sort of meta-analysis search for the tasks that most commonly activate that region, we get things like theory of mind and mentalizing, and same here for this peak down here in the cerebellum. So that was just a nice confirmation that our task, our stimulus, was provoking people to sort of engage in this type of cognitive thought. But again, what we're interested in is not so much what's common across the whole sample, but what varies with levels of trait paranoia. And when we run that analysis, when we look at, so we just did a median split, our paranoia measure, so we had a high paranoia group and a low paranoia group. And when we look for differences in ISC measure, the level of synchrony during story listening, we see 4 regions that come out as more synchronized in the high paranoia group than in the low paranoia group. And I won't read these to you in the interest of time, but several of them were regions that we know to be involved in sort of social cognition and processing. We took a lot of care to rule out sort of effects of just general cognition or memory or attention to the stimulus, and we're fairly confident that these are true differences in trait paranoia. And so given that we've identified these regions, we wanted to know which parts of the story they were responding to sort of more in the high paranoia group than the low paranoia group. And I'm going to kind of speed through this for interest of time, but what we saw here is that during moments that were particularly suspicious, sort of as marked by an independent group of raters, we saw more activity in the 2 regions that seem to be involved in social cognition from that previous contrast. So the left temporal pole and the right medial PFC were more strongly activated to these sort of suspicious events in the high paranoia group. And as a positive control for that, we didn't see any differences in that posterior SCS or TPJ region that was strongly synchronized in everybody. And then as a negative control, we also didn't see differences in left heschel gyrus or primary auditory. So this was an effect that seemed to be specific to those social regions, and they were more active again in that high paranoia group than the low. And so the presence of that naturalistic stimulus, that story, really allowed us to, again, get back to some sort of ground truth for the type of activity the type of information that's really driving activity in these regions differentially across individuals. So to sum up this last section, this is sort of an initial proof of concept study that we did, but we saw some interesting results such that we could use this ambiguous social narrative to evoke differences along this axis of paranoia. And so moving forward, we're really interested in fleshing this out a bit more, and so there's a whole lot of questions in terms of the best way to characterize these individual patterns of cognition. So which acquisition state should we be looking at? Rest is certainly powerful and convenient, but we're seeing some evidence that it may actually not be the optimal state, and we may want to sort of give people tasks, and that's sort of what I mean by this stress test. So we may get more bang for our buck, so to speak, or get more mileage out of data where we're actually perturbing the brain or sort of pushing it into a state that's especially one that's related to the traits that we care about. There's many different ways to analyze the brain data, obviously, so I talked about a few today, functional connectivity and inter-subject correlation. There's other ways that we could do this. Finally, I think a big area to think about is how we do the phenotyping. So what exactly are we looking for relationships to in the brain? And self-report and task performance are common ones. We're also increasingly interested in naturalistic behavioral capture, so for example, patterns of speech. And finally, again, as was alluded to in the talk this morning, I think longitudinal outcomes are going to be really critical for this work moving forward. So in conclusion, we can find robust and meaningful individual differences in brain function so that we can identify people from their patterns of brain connectivity, and we can use those patterns to predict cognitive behavior. We're seeing that brain state impacts in a fairly substantial way the measurements that we're making, so rest may not be the optimal state, cognitive tasks may be better. And finally, some preliminary evidence that we can directly manipulate brain state to draw out variation that we care about, and we're excited to extend this approach to additional axes of variation and additional brain states or scan conditions. So with that, I'd like to acknowledge my laboratory at NIMH as well as collaborators at Yale, and leave you with this final visual depiction of dangers of averaging across subjects. So thank you.